Hello and welcome to my students from the course Psych 2003 Physiological Psychology. I'm very happy to be partnering with you to explore the brain and behavior of physiological psychology. At the core of this course is the understanding, or maybe the assumption, that everything that we see as behavior, your moods, your decision making, how you feel at a particular time, whether you're hungry or whether you're full, all of those things are regulated by underlying biological structures. And of course, if we're talking about behavior, we'll be focusing primarily on the nervous system. Therefore, before we can go any further, we need to take the time to do a brief tour of the nervous system. In this lecture today, you'll meet some new terms, and it's very, very important that you begin to acquaint yourself with these terms, and these terms become very, very familiar to you. Because if we are to go forward, you need to have an appreciation of the nervous system, its structure, in order to understand its function. Before we go any further, let me just say this is an exciting new field of endeavor. In the past 10 to 15 years, this field has been exploding all across the world, and there have been tremendous strides being made. And it's really interesting to see this now, this discussion now coming to the Caribbean region. And I hope you find these lectures over the next few weeks as interesting as I do. So let us begin by exploring the nervous system, the structure of the nervous system. And we can say that the nervous system is divided into two divisions, or two components. The nervous system can be divided into the central nervous system and the peripheral nervous system. The central nervous system is made up of the brain and the spinal cord. And in the central nervous system, we also include the retina, the collection of receptors, rods and cones and nerves found at the back of the eye. The peripheral nervous system consists of sensory neurons that take sensory information from the periphery and bring it back to the central nervous system. And it also consists of motor neurons, which take commands from the central nervous system out to the periphery. You'll notice I've started to use a term already that should become very familiar to you, and that's the term neuron. A neuron simply refers to a nerve cell, and we'll explore the cells of the nervous system a little bit more in other lectures. So you can see in this diagram on the right that we have our central nervous system, which consists of the brain and the spinal cord. And coming off the brain and the spinal cord is our peripheral nervous system, which consists of both the sensory and the motor nerves. With that understanding, we can have a very simplified but useful picture of the nervous system. The nervous system then consists of the central nervous system, seen in the box on the right, which is known as the integration center. The integration center must receive information that it processes, and it receives this information via sensory input and sensory nerves. Once the integration center or the central nervous system has processed that information, it must send commands to the periphery or to the rest of the body, and it does this via motor nerves. And so we can say that the peripheral nervous system is like the action arm for the central nervous system. It receives information and it takes commands from the central nervous system out to the periphery. This is a very, very useful way for thinking about the nervous system, and it allows us to think about the different functions that the nervous system performs in this very simplified model. Now, we're going to focus primarily in today's lecture on the brain. So we're looking primarily at the central nervous system, and in today's lecture we're focusing not so much on the spinal cord but upon the brain. Now the brain is a really interesting organ. Actually, compared to most of the body, it's relatively small. It only weighs approximately 3 pounds. And 3 pounds represents about 2% of the average human's body weight. Yet despite it being such a small component of your body mass, it actually consumes between 20 and 25% of your daily oxygen and energy expenditure. So the brain then is a very, very small organ, or not very, very small, but it's a small organ, but it consumes a lot of energy. A lot of the body resources go to maintaining the brain and keeping the brain functioning and keeping the brain active. That immediately tells you about the important role that the brain has to play in sustaining life and in influencing behavior. Now, the brain is made up of two types of cells. I've already mentioned to you the cell that I think most people know about, and those are the neurons or the nerve cells. And it has been estimated that there are approximately 85 to 90 billion neurons inside of the average brain. Now, that's an amazing number, and that highlights the complexity of the brain and the challenge that man and neuroscientists have, and psychologists and physiological psychologists have, in trying to understand the brain. Because as we will see in subsequent lectures, not only are there 85 billion nerve cells, but these nerve cells talk to each other through synapses. And when you look at the number of synapses, there are literally trillions of synapses in the brain. In addition to the nerve cells, there's approximately an equal number of supporting or glial cells. These cells are increasingly being found to have an important role, not just to support the nerve cells, but also in brain function and behavior. And we will talk briefly about these in this lecture on cells of the nervous system. Now, the brain, due to its critical importance, is afforded a lot of protection by the body. The most obvious form of protection is the fact that the brain sits inside of the bony skull. And so therefore this bony skull, this bony chamber, offers protection to the brain. Once you move beyond the skull and the skin, you find that the brain is invested or covered by three membranes. And these membranes are known as the meninges. I'm sure many of you have heard of the condition meningitis. Anytime you see the term itis, that simply means inflammation. So if you hear the term meningitis, what is being described is there's an inflammation of the meninges of the brain. The membranes that cover the brain have become inflamed and infected. Of course, due to the proximity of the meninges to the brain substance, this can have catastrophic effects and is a condition that needs to be treated as a matter of grave urgency. Now, these three membranes have three distinct names. The outermost membrane, the one that's closest to the skull, is very, very thick and tough, and it's called the dura mater, and therefore it serves as a thick protective covering to the brain. Lining the dura mater is a much thinner membrane known as the arachnoid mater. Protruding from the arachnoid mater through the dura membrane are some small protuberances known as the arachnoid villi, and I'll explain what these do in a little while. But you can see them in the image to the right of the text. So far we say we have the dura mater, which is a thick protective covering. Lining the inner part of the dura mater is the arachnoid mater, which is a much thinner membrane. And then the third membrane is a very, very thin membrane that literally clings to and invests the brain tissue. When I say invest the brain tissue, you will notice the brain has a number of grooves. This pia mater literally dives down into the grooves with the brain tissue. And so wherever you find brain tissue, you're going to find pia mater. The space between the arachnoid mater and the pia mater is known as the subarachnoid space. So let me just repeat that for you. You have three membranes that cover the brain. They are known as the meninges. The outermost membrane is the dura mater. The middle membrane is the arachnoid mater. And the innermost membrane, which is almost like glad wrap, that literally clings to the brain, is known as the pia mater. Now, the brain is further afforded protection by the fact that the brain sits inside of a fluid. And this fluid ensures that the brain remains in a buoyant place. And this fluid is known as the cerebrospinal fluid. And that fluid is found in the subarachnoid space. So you will remember that I told you that between the arachnoid membrane and the pia mater is a space, and this space is called the subarachnoid space, and it's filled with cerebrospinal fluid. This cerebrospinal fluid circulates around the surface of the brain, but it's produced in some cavities in the brain known as ventricles by an area called the choroid plexus. And from these ventricles, it circulates throughout the brain. And then it is reabsorbed and taken back into the bloodstream for further processing in the body. This cerebral spinal fluid, not only does it provide buoyancy to the brain and help protect it, but it also provides a stable chemical environment that's important for keeping the homeostatic environment of the brain constant, and is very important for clearing waste materials from the brain. All of these are aspects of the brain and aspects of the central nervous system that I'm going to ask you to do a little bit more reading on. This lecture simply serves as an overview to introduce you to different structures and different aspects of brain anatomy and function. So let's summarize what we've seen and learned about the brain, the cerebrospinal fluid, the meninges, by this little video which was taken from YouTube.
The cerebrospinal fluid, or CSF, is the liquid that bathes the central nervous system, the brain, and the spinal cord. The CSF ensures the protection of the central nervous system against mechanical shocks and vibrations. It plays an important role in the regulation of intracranial pressure. It participates in the cerebral metabolic processes by regulating the exchange of substances and provides many other functions as well. The CSF circulates in two different compartments. The first is the inner brain ependymal cavities, or ventricles. The second is the compartment surrounding the central nervous system. It corresponds to the subarachnoid space. The CSF is secreted by the choroid plexus. Clusters of capillary blood vessels located in the ventricular system. The ventricular system consists of 4 ventricles connected by many channels. It is connected to the central canal of the spinal cord. There are 2 lateral ventricles occupying the center of the cerebral hemispheres. Both are connected to the 3rd ventricle at the center of the diencephalon through the foramina of Monroe. The 3rd ventricle is connected to the 4th ventricle in the brainstem by the aqueduct of Sylvius. The CSF will leave the 4th ventricle through the foramen of Magendie to fill the subarachnoid space between the pia mater and the arachnoid. The CSF is then absorbed at the top of the skull by the arachnoid granulations of Pacchioni.
So there we had a nice simple video from YouTube that went over the structure of the ventricular system of the brain, pointing out to you that there are two lateral ventricles, there's a third ventricle and a fourth ventricle. Those granulations of Pacchioni are the arachnoid villi that I mentioned to you before. So let's press on with our tour of the brain. Well, one way of understanding the anatomy of the brain is the fact that we can divide the brain into three regions or three areas known as the forebrain, the midbrain, and the hindbrain. These divisions are primarily due to or originate from the embryological development of the brain as the brain was developing in utero. And let's look a little bit at each part of the brain using this nomenclature or this classification. So the hindbrain links the brain to the spinal cord. It's found at the base of the brain, and it consists of the lower part of a key phrase, the brain stem, and the cerebellum. The brain stem would represent not just the hindbrain, but the hindbrain and some other structures. And so we can say that the hindbrain consists of the medulla, the pons, the cerebellum, and the reticular formation. So the hindbrain consists of four key structures that I'd like you to know about for this course: the medulla, the pons, the cerebellum, and the reticular formation. Now the medulla is very, very important, as is the hindbrain and the brainstem in general, because this area of the brain is important for regulating a number of our vital body functions, such as your heart rate, your blood pressure, your respiratory rate, how you regulate temperature. Many of these processes are regulated in and around the hindbrain. And the medulla in particular is important for controlling your heart rate, your blood pressure, and your breathing. Just above the medulla is the pons, and the pons also has a key role to play in the aspect of regulating breathing. And it also contains a number of cranial nerves that are important for hearing, equilibrium, and taste. Now this part of the brain, the brain stem and the hindbrain region, is the part of the brain, as I said, that keeps a number of your vital bodily functions going. When you talk about brain death, this is the area that is ultimately no longer functioning and allows somebody to be termed brain dead. You can have a catastrophic injury and the larger parts of the brain, like the forebrain, are no longer working, but your brain stem can still be working. But once the brain stem has stopped working, we describe somebody as being brain dead. Within the structure of the hindbrain and the brain stem is a diffuse collection of neurons that are responsible for arousal. And these neurons are known as the reticular formation. And these help you to stay alert and awake. Very, very interesting area of the brain that not a lot is known about, but we do know it is critical for arousal. Now, behind these structures is a region of the brain called the cerebellum, also known as the little brain, because it looks just like the brain with all its undulations and convolutions, but obviously it's much, much smaller. It also, interestingly, has about half all the neurons for the entire brain. So it's very, very densely packed with nerve cells. And this region of the brain is vital for motor planning and motor coordination. Persons who have cerebellar disease or cerebellar injury have difficulty coordinating their movements. The cerebellum is also very important for memory as it applies to skilled movements and learning to play the piano or learning to ride a bike. And we'll talk about that in our lecture on learning and memory. And so the hindbrain consists of these four structures: the medulla, the pons, the cerebellum, and the reticular formation. If we move on to the midbrain, and we're not going to spend much time on the midbrain, but we can see it's the region just above the hindbrain that I'm highlighting here. Inside of this diagram, I want you to note we now have the full extent of the brainstem, which consists of the medulla, the pons, the midbrain, and the thalamus. Now, the midbrain is an area that we're not going to focus on much, and it is important as a relay center for vision and hearing. A lot of the pathways involved in vision and hearing pass through the midbrain, and also it has a role to play in motor planning. But for the purposes of this course, we won't be spending too much time looking at the midbrain, but it's important that you appreciate that it exists and some of its basic functions. What we do want to focus most time and attention on is the forebrain, or the largest area of the brain. And you can see the forebrain consists of the cerebrum, which is this large region here. It consists of a structure called the thalamus, which is considered the sensory gateway of the brain. Almost all sensory information has to pass through the thalamus before it goes to the rest of the brain. And it consists of the limbic system, which is very, very important for processing our emotions. And we'll see that in our lecture on the emotions. We're going to focus on the cerebrum, which is the largest division of the forebrain and the brain in general. And the cerebrum is divided into two hemispheres, each of which is divided into 4 lobes. And each hemisphere is connected to the other via a tract of white matter, the corpus callosum. Now, I introduced another term to you there, white matter. What do we mean when we say white matter? Well, histologically, when you're looking at the cells of the brain, you can see that there are 2 types of regions. There are regions where you see the cell body, and these tend to appear gray in the brain. But then there are regions where you see these long processes called axons that come off the cell body. And these refer to the tracts where information is being transferred from one area to another. And these appear white on the brain. And so this is called the white matter. And the corpus callosum represents the areas or the tracts in which the left hemisphere of the brain communicates with the right hemisphere of the brain. Now, if we look at the cerebrum in a little bit more detail, we can see that there are a number of grooves upon its surface. And there are some key grooves that I just want to mention to you. The right and left hemispheres are divided by this long sagittal groove that runs right along the midline. And this groove is known as a longitudinal fissure. The cerebrum is separated from the cerebellum by another very deep groove known as the transverse fissure. And there's a third deep groove that is very, very large, and this is called the sylvian or the lateral fissure. And this separates the temporal lobe from the frontal lobe and the parietal lobe. So let me go through that in a different way. Let me remind you that there are 4 lobes of the brain. To the front of the brain, we have the largest lobe known as the frontal lobe. To the back of the brain, we have the smallest lobe known as the occipital lobe. To the side of the brain, we have the temporal lobe. And the 4th lobe is the parietal lobe. The brain substance is separated by 3 key large fissures. Separating the brain into 2 hemispheres is the longitudinal fissure. Separating the cerebellum from the cerebrum is the transverse fissure. And separating the temporal lobe from the frontal and the parietal lobe is the sylvian or the lateral fissure. As I mentioned at the beginning, it's very, very important that these terms do not intimidate you, and that you take the time to learn these terms and understand these terms, and that these terms become second nature to you. It may be like learning a new language at this point, but remember, if you keep practicing, it will become very, very easy as you go forward. So if we take a section through the cerebrum, we can see that there's an outer thin layer known as the cerebral cortex. And I'm highlighting that with the mouse here. And this layer is approximately 2 to 4 millimeters thick. And this is where all the cell bodies of the cerebrum are found. Below there you have the white matter. And I already explained to you the difference between the gray matter where the cell bodies are found and the white matter where we have the tracks or the axons coming off from the nerve cells. You will also notice that the brain has all of these convolutions. The elevated ridges that you find on the brain of brain substance are called the gyri. The grooves are called the sulci. So let's say that again. The elevated ridges are called the gyri, and the grooves are called the sulci. Gyrus for singular, sulcus for singular. So let's look a little bit at more detail at the lobes of the brain. Remembering that this is just an introductory tour, we're not going to go into a lot of detail, but in other lectures we'll explore these things in a little bit more detail. So at the back of the brain is the smallest lobe, the occipital lobe. And the occipital lobe is known primarily for one thing: visual processing, the processing of visual information. If you have damage to your occipital lobe, it can lead to complete or partial blindness. Now, one would wonder, well, why do you need such an area simply for processing vision? Well, vision is one of our most important senses, and it's not a simple sense. It is very, very complex. And so different regions of the occipital lobe and different neurons in the occipital lobe are responsible for processing different aspects of vision, including the size of objects, the color of objects, how light is reflecting of objects, are the objects moving, and what are the three-dimensional structure of the objects. And scientists have spent a lot of time trying to understand how we process vision as a model generally for how the brain processes information. The next lobe we're going to talk about is the frontal lobe. You can see in the image here, the frontal lobe lies to the front of the brain, hence its name. And it actually is the largest lobe in the brain. Now, to discuss the frontal lobe, I'm going to introduce you to one of the most famous clinical cases in physiological psychology or behavioral neuroscience. Before we had the wonderful imaging techniques that we use to explore inside of the brain, one of the main ways in which we could understand the relationship between brain and behavior were rare clinical cases. Cases in which we could clearly identify a structural problem in the brain and then look at how that manifests in changes in behavior. And one of the most famous cases that is found in almost any psychology textbook that you may have heard about already is the story of Phineas Gage. So let's just look at this little video and listen to a brief account of Phineas Gage.
Phineas Gage was once an ordinary railroad foreman. He was a happy, healthy, and friendly person until a freak accident caused him irreparable brain damage. Now, Gage is remembered as a famous medical mystery. The accident that drastically changed his life and personality also changed contemporary understandings of the human brain. The horrific accident occurred in 1848 when Phineas Gage was clearing rocks from a new railroad's future path. He was placing gunpowder into the cracks in rocks and then detonating them to clear them. One day, he made a mistake with his explosives. Gage forgot to cover the gunpowder he was patting down with sand. When he tried to pat the gunpowder into the rocks, it exploded and launched a 3-foot-long metal pole through his skull. The projectile dragged parts of both his frontal lobes along with it. Despite the gruesome injury, Gage was relatively unaffected by the accident. Some reports say that he convulsed for the first minute after the impact, but many believe that he never actually lost consciousness. Regardless, he was undoubtedly conscious about 1 minute after the impact. Gage sat up and actually spoke to his fellow railroad workers. When they wheeled him to a doctor, he insisted on being given his time book to continue doing his job as a foreman. While Gage's determination to continue working despite his terrible injury may seem odd, the effects of his injury were even stranger. The open brain wound healed after only 10 weeks, and Gage was able to count, sing, feed and dress himself, and make sense of information that was given to him. Some aspects of Gage never fully recovered, however. His personality was completely different. His old friends could not tolerate his newly inconsiderate attitude, inexplicable temper, and constant profanity. Soon, he was fired from his job for being nasty to co-workers, people who he had worked well with just 10 weeks before. Gage was so out of control that men kept him away from women and children because he was considered too vulgar for them. Even though he had physically healed, something was still wrong. As his doctor said, Gage was no longer Gage. 11 and a half years after the accident, Gage died. Unfortunately for the medical community, no autopsy was conducted on his unique brain. Years later, his body was exhumed with permission from his family, and scientists studied his skull. They saw approximately where the rod traveled through his head, and did their best to understand the damage with their limited understanding of the brain. The case study became an essential part of the debate regarding the function of the brain in the mid-1800s. Two prominent schools of thought existed. Ironically, both used Gage as support for their theory. One group believed that the brain was connected by a vital force. Due to this vital force, any part of the brain could perform any of its functions. Therefore, someone like Gage could still think and behave like everyone else, despite having lost a large piece of his brain. The other group of brain theorists, commonly referred to as the localizers, thought that the brain had different parts that localized specific functions. They pointed out that Phineas Gage's personality was never the same after the accident. The localizers believed that the damage to the brain caused a lasting change in Gage's personality. Ultimately, both competing schools of thought were integrated into contemporary brain theory. The brain does have specific localized functions. Modern research has shown that motor functions are certainly localized to specific areas. Localizers, however, underestimated the interconnectedness of the brain. The brain has trillions of synapse connections, and many human functions, such as memory, are impossible to localize to only one area in the brain. Recent studies have shed more light on Phineas Gage's specific condition. Damage to the frontal lobes has been shown to cause behavior spontaneity. Patients with frontal lobe damage often have difficulty interpreting information from their environments, taking risks, and complying with the rules. Especially with injuries that damage both lobes, a person's personality is often drastically altered. These recent studies help explain the effects of Phineas Gage's injury. Still, scientists wish to better understand Gage's disorder and the underlying brain science that could explain it.
So now you've met Phineas Gage. Here's another picture of Phineas Gage that you might have seen in the video. Him holding the rod that maybe went through his head. I don't know if it was the actual rod. This middle image is his doctor who took care of him, Dr. Harlow. And this image here to the right is a reconstruction that was recently done showing the path of the rod through the left side of Mr. Gage's brain and how it would have primarily damaged the left frontal area and destroyed a lot of the tracts in that region. Unfortunately, a lot has been written about Mr. Gage, and his symptoms have been perhaps greatly exaggerated after the injury. But we do know there did seem to be some change in personality. And this basically led to the theory that different parts of the brain have different functions, as was mentioned in the video. And now we understand that the frontal lobe is believed to be involved in decision making and reasoning, social interactions, personality, emotional processing, and memory formation. So for example, persons who have dementias that affect the frontal lobe and the temporal lobe, eventually it affects their personality. So this is very, very interesting for us inside of this course, because this is providing evidence that some core aspects of who we are and our behavior are not just random and not just strange, but they actually have underlying biological substrates to them. And if we can understand these substrates, scientists are pushing the boundaries to consider, can we manipulate these substrates, primarily for useful reasons, but of course we know there will always be people who are thinking how they can use these things for nefarious reasons. And this of course has opened up a whole new realm of endeavor known as neuroethics. As the boundaries on brain science get pushed, do we need to now consider the ethical implications of what scientists are actually doing? So let me just repeat, the frontal lobe now is supposed to be, or is known to be, involved in decision making and reasoning, social interactions, personality, emotional processing, and memory formation. Of course, these are big, broad areas, and the exact way in which the frontal lobe works in these areas is a matter of intense scientific investigation at present. The frontal lobe is also very important because of its critical role in motor function. Now, one of the main grooves in the brain is called the central sulcus, and it separates the frontal lobe from the parietal lobe. Now, there's a strip of tissue called the precentral gyrus that sits right in front of the central sulcus. And this area of tissue is known as the primary motor area. And it is responsible for simple movements of the body. Now, on this slide here, there's a little cartoon from the far side. Here we have some neurosurgeons, and they're doing brain surgery. And one of them says, "Whoa, that was a good one. Try it, Hobbs. Just poke his brain right where my finger is." And then you see his foot, one of his legs contracts and pops out. So here the far side cartoonists are making fun of something we know well about the brain. That if you stimulate this precentral gyrus, this primary motor area, you can cause discrete movements of different parts of the body about a single joint. And what has been discovered is that each area of the precentral gyrus, that primary motor area, controls for a different part of the body, as demonstrated by this image on the right. So there's a set of neurons that control the foot. There's another set of neurons that control your hip. There's another set of neurons that control your trunk, and some that control your hand and your face. Now I want you to look at this image on the right for a little while. This image is known as the motor homunculus. And I want you to think about if you notice anything strange about this image, or if anything strikes you about this image. Take a few seconds. What do you notice? Well, I think we've said already that different areas control different parts of the body. But one of the most striking things is that the areas that control the face and the hand appear much larger than the areas that control the legs and the trunk. On your body, the legs and the trunk would make up the majority of your body or your body mass. But they seem to be under much less control than the areas responsible for the hand and the face. What this actually says is there are a lot more neurons or a lot more nerve cells used to control the hand and the face than the trunk and the legs. And this reflects the fact that the hand and the face can make much smaller granular movements. The hands and the face can make much more specific movements, and therefore they require much more control by the brain to make these distinct movements. Hence their large regions being expressed on the primary motor cortex. Now this next image is very funny because this is what your brain sees. So this is you on your brain. This is the primary motor cortex, and it's been a reconstruction of what you would look if you looked at your brain, or if you constructed you based upon your brain. And you can see again, the largest areas of the brain are devoted to the hands and to the face, particularly the tongue and the lips, that are very, very important for processing different information and also speech. The sensory areas of the brain are also very similarly constructed. And so your brain actually represents you in a very, very different way from how you actually exist in real life. So that's the frontal lobe. Very important for personality, social decision making, but also important in other features like motor control. Now behind the frontal lobe is the parietal lobe. Remember I told you about the central gyrus. Now the parietal lobe is very, very important for processing sensory information. So right in front of the central gyrus, we have the precentral gyrus, which is the primary motor strip. But right behind the central gyrus, we have the postcentral gyrus, which is the primary somatosensory strip. This area is responsible for processing sensory information. And if we go to our previous slide, just like the motor cortex, each area of the primary somatosensory area codes for a different part of the body. So information from the hands goes to one part of the primary somatosensory area. Information from the legs goes to another part. Information from the lips and the tongue goes to another part. And in this way, the brain is able to understand that when certain parts of it stimulated, that stimulus is being detected by receptors found on the hands or the tongue or the trunk. The other main area of sensory processing that we find in the parietal lobe is the gustatory center, which also processes taste. This arrow here highlights very clearly the postcentral gyrus and the primary somatosensory area that I was just telling you about. Now, because the parietal lobe is very involved in integration of sensory processing, damage to the parietal lobe can create some strange phenomena. And one of the strange phenomena that you might like to read about is something called hemi-neglect, where it seems that persons seem to not register that half of their body actually exists. And this can be so bad and so devastating that a gentleman with this syndrome can actually wake up in the morning and literally only shave half his face. If such a person is asked to copy an image like this one, as you can see, they only seem to draw one side of the image. And if they make a spontaneous drawing on their own, again, they only seem to give detail to one side of the image. Very, very strange lesion, as many brain lesions are, but actually point to the criticality of different regions of the brain and what they do. And in this case, the parietal lobe is very, very important for the processing and the bringing together of different parts of sensory information. So we have looked at 3 lobes so far. We've looked at the occipital lobe to the back of the brain, and we've said the occipital lobe is important for the processing of sight. We've looked at the frontal lobe to the front of the brain, and we've said the frontal lobe is very important for personality, decision-making, memory formation, emotional regulation. We've also seen that frontal lobe has a key role to play in simple motor movements and motor control. We've just looked at the parietal lobe, and we've seen that the parietal lobe is important for sensory processing, especially the processing of taste and touch. And the last lobe we'll look at is the temporal lobe, which is located to the side of the brain. The temporal lobe is known to be important for the processing of auditory information, your hearing, the processing of speech and language, and also for memory formation and retrieval. Now, these first 2 issues, auditory information processing and speech and language, we'll examine in a lot more detail when we consider how does the brain involve itself in communication. So we're going to come back to the roles of the temporal lobe inside of these functions in another lecture. So let's just say for now that the temporal lobe is involved in auditory information processing, the processing of speech and language, and memory formation and retrieval. And so this ends our brief tour of the brain. I thought I would end by taking another video from YouTube. And as I take these videos and play them in this lecture, I want to encourage you to go online and you use the number of resources that are available to you on the internet, as well as your textbook, to find out more about the brain. So we're going to just look at this last video, which just talks a little bit about some neuromyths before we close. So let's enjoy this video.
Get the book at asapscience.com. The brain is amazing and complex, but our lack of understanding has led to a great variety of myths throughout the years. Like, bigger is better. While this may be true for some other organs, you only need to look at a sperm whale's brain, which is nearly 6 times larger than that of a human's, to see this myth fall apart. Yes, they're certainly intelligent animals, but their cognitive skills pale in comparison to our own. Had a fun night out? Don't worry, alcohol doesn't actually kill your brain cells. Although it does cause damage and creates problems conveying messages between neurons, the damage is mostly reversible. Similarly, drugs don't actually create holes in your brain. Though some can permanently change your brain's functionality and structure, the only thing that can put a hole in your brain is physical trauma. Good thing you have 100 billion brain cells, or not. Surprisingly, it wasn't until 2009 that scientists discovered the true number to be closer to 86 billion. Sure, it may seem like a small difference, but those 14 billion neurons are equal to the size of an entire baboon brain. To further put those numbers in perspective, consider that 1 million seconds is 12 days, while 1 billion seconds is 31 years. So that 14 billion neuron difference may not be so small after all. Regardless, we can't use them all, right? Most movies and sci-fi books have us believe that humans can only use around 10% of our brain, which is absolute nonsense. Thanks to modern brain scanning technology, we know that we use the whole thing all the time. Not necessarily all at once. When you're walking, for instance, the parts of the brain associated with movement are more active than other areas. However, there is no part of the brain that simply doesn't do anything. It represents 3% of the body's weight and uses 20% of its energy. That's a busy brain. But we generally use one side more than the other, right? Nope. While you may have been convinced that if you're more logical or analytical, you're more left-brained, and if you're more creative or intuitive, you're more right-brained, it's simply not true. While different sides of the brain are indeed used for specific functions, for example, language uses the left hemisphere while the right hemisphere helps to read emotion, studies on thousands of individuals have found no evidence of a left or right dominance in individuals. In other words, you use both sides equally. And you don't actually have 5 senses like you were always told. In fact, you have a lot more. like nociception, which is the sense of pain, and proprioception, the sense of how our bodies are positioned, not to mention the sense of balance, temperature, and passing of time. Scientists at GE's global research centers are collaborating with top researchers and institutions to uncover new insights about the brain's functionality. If you're interested in debunking more myths or understanding more about the brain, you can follow their progress at GE Reports as they work together to crack the brain's greatest mysteries. Special thanks to GE for supporting this episode of ASAP Science. And subscribe for more.
So, just a fun little video at the end to talk about some common myths concerning the brain and neuroscience. Let me just make one statement about that last point, that we have more than 5 senses. Traditionally, the way we approach that in neuroscience and physiological psychology is the fact that we have 5 basic senses, but each of these senses is separated into what we call submodalities. So if you think about something like sight, that's the basic sense of vision, but then you can have different colors inside of that separation. And primarily, you talk about seeing in black and white and seeing in color. If you think about something like touch, we can divide that into different types of touch, like crude touch, such as tickle and itch, or fine discriminating touch that allows you to identify a particular object. And so that's what they were talking about right at the end there for that last point. So that ends our tour of the brain. I hope you found it useful. As I said, this was just an overview. I'm now going to ask you to make sure that you spend some time reading through these things inside of your textbooks, and we will discuss these things further as we go forward in the lecture. I want to thank you for listening, and I hope you enjoyed our brief tour of the nervous system today.